Okay, so welcome to this second video in which we are looking at the G-inhibitory pathway. Okay, so we have so far got to the stage where we have actually made our alpha IO bonded to GTP subunit, and we now want to see how it's going to act on these, specifically on these two adenylyl cyclases, adenylyl cyclase 1 and adenylyl cyclase 8. And basically what I told you in the previous video was that it works by stopping these two adenylyl cyclases from being activated by calcium. Okay, so let's see how that works. So if we start off with adenylyl cyclase 8, because it's the more difficult one, then basically if we look at the structure of adenylyl cyclase 8, if this is the phospholipid bilayer here, then basically adenylyl cyclase 8 has an N-terminal domain here, which has a very special domain called the calmodulin recruitment domain, which we'll look at in a moment. Then it has 6 membrane-spanning alpha helices clustered together in a domain known as transmembrane domain 1, or TM1 for short. Then it has a loop connecting this transmembrane domain 1 to a second transmembrane domain, which again has 6 membrane-spanning alpha helices, and this second transmembrane domain is called transmembrane domain 2. And then finally you have a carboxyl terminal domain, which is called the C2 domain. So here's C1, this is the C1 loop, and C1, which is the loop between transmembrane domain 1 and transmembrane domain 2, has 2 domains, C1a and C1b, and the C2 domain over here, which is this carboxyl terminal domain, it has also 2 domains, which can be divided up into C2a and C2b. Okay, and I said at the beginning that this domain over here is known as the calmodulin recruitment domain. Okay, right. So if you want to activate this adenylyl cyclase 8, what you need to do is actually make a functional catalytic enzyme, basically, an enzyme which is going to take in ATP and break it down into cyclic AMP and pyrophosphate. And the way in which you create a functional enzyme is you create it out of this C1a portion here and this C2a portion here. So in order to create a functional enzyme, you have to dimerize C1a with C2a. So you need to put this portion together with this portion, and together they make a functional catalytic enzyme which is going to catalyze that adenylyl cyclase reaction. Okay, so the way calcium triggers this is as follows. Adenylyl cyclase 8 does not actually have a calcium sensing domain in its internal structure, so it relies on a different, an external calcium sensor, and that external calcium sensor is calmodulin. So let's quickly go over calmodulin. So calmodulin has 2 lobes. It's often drawn in cartoons like this. It has 2 lobes, an N lobe and a C lobe. Okay? A C lobe. And the clobe, that looks like C lobe. I'll put a dash in there to denote that they should be apart. Okay, so each of these 2 lobes of the calmodulin has 2 binding sites for calcium. Here they are. So these calcium binding sites are specifically what are known as EF hand domains, which is basically just a very specific shape of a polypeptide that looks kind of like this. You have a loop basically like that. And the amino acids in this portion of the polypeptide that makes up this loop are all acidic amino acids, which means that they donate protons. Now, when they donate protons, they gain a negative charge. So all of these residues in this portion of the protein gain a negative charge, and that allows them basically to coordinate a calcium ion in the center, basically there. And these EF hand domains often occur in dimers, known as EF hand dimers, like so. And indeed, this is what happens in these lobes of calmodulin. You get 2 EF hand domains next to each other in a dimer, and therefore 2 calciums bind, and you have 2 calcium binding sites here. Okay, right. So, when calcium binds to this this calmodulin molecule, and by the way, calmodulin, when it doesn't have any calcium bound, is known as apocalmodulin, or it's often abbreviated to ApoCAM, so apocam. CAM, like that. And it's often done in that way, where you have the capital C, the lowercase a, and the capital M. Okay, so that's apocalmodulin. When calcium binds, what happens is that the calmodulin changes its conformation. Firstly, these two lobes move further apart, so it takes on a shape more like this. And also, this linker between the two loops, it was linear, but now what's going to happen is that it's going to take on an alpha helical structure, like so like this spring that I'm drawing here. Okay, right, and what will happen is that alpha helix generally wraps around proteins, and that's how the calcium calmodulin complex, as it's called now, is going to interact with proteins. So this is a calcium calmodulin complex. Okay, and there are two calciums bound to each of the lobes, one in each of the calcium binding domains. Okay, and calcium calmodulin complexes are often denoted Ca2CAM for short, so calcium calmodulin. So there should be calcium ions in each of these binding sites, basically, like so. Okay, so the way adenylyl cyclase 8 is activated is that apocalmodulin initially binds to this calmodulin binding domain over here. So you get apocalmodulin binding there. And when calcium goes up, the calcium binds to the calcium binding sites on the two lobes of this apocalmodulin. And when it does, it changes conformation to this calcium calmodulin complex form. And what also happens is that the calcium calmodulin complex no longer remains bound to this calmodulin recruitment domain. Instead, it's transferred onto this C2b portion. So what happens is that when this apocalmodulin becomes a calcium calmodulin complex, the calmodulin recruitment domain gives the calcium calmodulin complex to C2b. Okay, so here's our calcium calmodulin complex here, and I'll put little orange dots to denote that it's got calcium in it. And when you have calcium calmodulin complexes bound to this C2b portion, it promotes the dimerization of C2a with C2b, and you get the active enzyme being formed. Okay, and when that happens, the active enzyme is going to take in ATP and it's going to break it down into cyclic AMP and inorganic and pyrophosphate, and you'll get cyclic AMP produced. Okay, so we'll now look at adenylyl cyclase 1 and how it's activated by calcium binding to calmodulin again. Adenylyl cyclase 1 does not have its own intrinsic calcium sensor. It relies on calcium sensors in the form of calmodulin. So, okay, here is adenylyl cyclase 1. It's got the same structure as adenylyl cyclase 8, and again, it's activated in the same way as adenylyl cyclase 8, i.e. you need the dimerization of this C1a with this C2a region in order to get an active enzyme. Okay, so here's C1a here, here's C1b, here's C2a over here, and here's C2b. Right, okay, so what's different in the case of adenylyl cyclase 1 is that you don't have the calmodulin recruitment domain. Instead, what happens is that apocalmodulin binds to C1b here. So there's the apocalmodulin bound to C1b. Now, when the calcium goes up in the cytoplasm, this apocalmodulin is going to turn into a calcium calmodulin complex, and when it does, it changes the conformation of this C1b region, and that promotes the dimerization of C1a with C2a in order to form an active enzyme, basically. So here we are. Okay, so I'll draw that happening. Okay, so what's going to happen is that it's going to change conformation in this way, like so. And I'm drawing it again because it's valuable when we come to understand how the alpha I with GTP bound to it is going to inhibit this process. Okay, so here's the transmembrane domain 2, and then you have this dimerization now of C1a with C2a. And I realize that I have stretched C2a out rather a lot, but yes, that's just the picture. It doesn't really stretch. Okay, so this is C1a here, but I'm not going to label it because I need to just get this calcium calmodulin in C1b here. So this is the calcium calmodulin complex now, bound to C1b in there. And hopefully you can get from my drawing that I have dimerized the C1a domain here with the C2a domain here, and now that's an active adenylyl cyclase enzyme, and it will take in ATP, it will break it down to cyclic AMP and pyrophosphate, and you'll get a rise in intracellular cyclic AMP. Right, okay, so those are how you activate adenylyl cyclase 8 and adenylyl cyclase 1 using calcium. Right, so now what we need to do is have a look at how these alpha I/O GTP subunits block this. And basically what alpha GTP I/O GTP subunits do is they come in and they bind to the C1 region of the adenylyl cyclase, and they also bind to the C2 region, and they hold them apart basically. So if I show you what they do, let's go on to the other side. So if we draw another adenylyl cyclase here, so here's the transmembrane domain 1, here's C1, transmembrane domain 2, and here's C2, okay? And here's the phospholipid bilayer there. Right, basically, what the alpha I/O GTP subunit does is that it binds, and here's the GTP here, it binds to both the C2 region of the adenylyl cyclase over here, so this is the C2 region, and it also binds to the C1 loop between transmembrane domain 1 and transmembrane domain 2. And in that way it holds them apart and stops the C1a portion here from dimerizing with C2a over here. And therefore you can't get the formation of the active adenylyl cyclase enzyme, even if calcium tries to activate it. This alpha I/O GTP subunit is holding the C1a and the C2a apart, and therefore they cannot dimerize. So it inhibits adenylyl cyclases by stopping their activation, and it's specifically stopping the calcium activation of the adenylyl cyclases. And the two adenylyl cyclases which are famous for being calcium activated are adenylyl cyclase 1 and adenylyl cyclase 8. Okay, right, so when we stimulate our GPCR, we get the formation of this alpha I GTP subunit, and it is blocking the activation of these adenylyl cyclase 1 enzymes and these adenylyl cyclase 8 enzymes. It does block the activation of certain other ones, but adenylyl cyclase 1 and adenylyl cyclase 8 are the main ones which are blocked by alpha I/O GTP. Okay, right. So now what's going to happen is that the amount of cyclic AMP you're producing is going to go down, basically. So cyclic AMP production goes down. So if cyclic AMP production goes down, well, there are phosphodiesterases in the cytoplasm which are breaking cyclic AMP up. And their activity, no one's told them to stop working, so they continue working on at the same rate. So now you've had a decrease in production, your removal of cyclic AMP remains the same, so overall the equilibrium is going to break basically, and you're going to overall remove more cyclic AMP than you're putting in, so cyclic AMP concentration in the cytoplasm is going to go down. Now cyclic AMP is responsible for activating protein kinase A. Okay, so let's have a little discussion of type 2 protein kinase A, but we'll do that in the next video.